Good evening, everyone. I'm Andrea Bodner, the Donald G. Combs Science Director at the Gloucester Marine Genomics Institute, and it's my pleasure to welcome you to another episode of the GMGI Science Hour. Tonight, we welcome Ryan Phelan, who's Executive Director of Revive and Restore, who will be discussing their work bringing biotech solutions to wildlife conservation. But before the presentation begins, I want to share a little bit of background about GMGI, especially for those of you in the audience who may be new to us. And tonight I'm going to do this by sharing a brief video with you about our organization. The oceans are facing tremendous challenges, from climate change to plastic pollution. We're seeing tremendous change even in our own backyard.
GMGI is a nonprofit that addresses critical challenges facing human health, the oceans, and the environment through innovative scientific research and education.
We pursue three distinct but complementary strategies that braid brilliantly together. The first is our research. We conduct marine research powered by genomics. Our education initiative is providing a career launchpad for local high school graduates who are struggling to find opportunity. And the third is our science community initiative, where we are working to create conditions to attract biotechnology and life sciences companies to Cape Ann.
With the culture and economy of Gloucester deeply rooted in maritime history, it's the perfect location.
We have a remarkable amount of talent, capital, and innovation happening all at once. Companies that have come here have realized great support from the state, the city. They've witnessed a robust, capital-efficient industrial infrastructure. Gloucester is becoming a very popular place to do business.
Our science strategy is based on a platform of advanced molecular biology and genomic technologies to address questions related to oceans and human health with three focus areas: biomedicine and biotechnology, ecosystem function and health, and fisheries and aquaculture.
What's really exciting about GMGI is the way the technology has progressed is we're on the cutting edge, and that will allow us to answer these really complex and interesting questions.
GMGI is a really incredible place to work. Not only do we have this state-of-the-art research facility right on the water, we have a really incredible team of people who have a broad range of backgrounds and knowledge.
The Gloucester Biotechnology Academy aims to democratize access to the life sciences and to train people to fill the important critical roles as professional laboratory technicians.
For the Academy, I was kind of lost in my own path. I had always enjoyed science in school growing up, but it was just something that I always felt was out of reach. The Academy and the education I received has really given me a second chance. My time at the Academy was spent a lot in the lab doing hands-on training, and that had really prepared me to get out in the workforce. Now I work for a company called Elevate Bio in Waltham, Mass. I am an associate scientist there in their next generation sequencing core.
They come out of this with very specific skills, practical skills, intuitive skills that are very important.
It uncovers their love of science, their passion for science. You can see the change in their level of confidence.
Coming out of the academy, I have more hands-on experience than a lot of my colleagues that did 4 years in college. My internship then led me to a full-time job for a whole year at Dana-Farber. And then after my internship and my year there, I actually joined a private biotech company where I've grown tremendously. It's a small commitment that unlocks life-changing opportunities.
We've graduated 80% of the students who we've enrolled over the past 6 years. 90% of our graduates are working in life sciences or have enrolled in college to study science.
To be able to take young people who have had very little STEM education and be able to give them a huge opportunity for career growth, to me, is very amazing and special.
We're changing the trajectory of the community, both through bringing science to the community, making new discoveries that will have an impact on fisheries, on human health, and also changing the trajectory of the students we train at the Academy.
We are small but mighty. You would never know that we are a team of under 30 people when you see the groundbreaking research being conducted in our labs and the transformative education experiences being provided at our academy. And we are just getting started.
If you'd like to learn more about GMGI, please feel free to reach out to us. Now to introduce tonight's speaker. Ryan Phelan is the co-founder and executive director of Revive and Restore, and works with world's leading conservation biologists and molecular biologists to develop pioneering projects that apply cutting-edge biotechnologies to seemingly intractable conservation challenges. She has organized landmark workshops bringing together global experts to identify the challenges facing endangered species and what genetic tools can be used to help save them from extinction. In 2021, Ryan captured the TED stage in Monterey with her talk titled The Intended Consequences of Helping Nature Thrive. And in 2023, Ryan reflected on 10 years of progress at Revive and Restore with a seminal presentation at the Long Now Foundation titled Bringing Biotech to Conservation. Ryan is a serial entrepreneur, active in the for-profit and the nonprofit worlds. For 2 decades prior to her conservation work, she was a leader in patient-oriented healthcare and was the founder and CEO of 2 healthcare companies focused on empowering consumers, DNA Direct and Direct Medical Knowledge. So please join me in welcoming Ryan to the GMGI Science Hour. And if you have any questions throughout the presentation, please use the Q&A function at the bottom of your screen, and Ryan will answer those questions at the end of the presentation. So take it away, Ryan.
Well, thank you, Andrea. I have to say, I love seeing that video. Number one, I need to be recruiting some of your graduates. I mean, the training that you do is so in line with our nonprofit Revive and Restore. So I will try to get through as much of this deck as I can on Zoom so that we can have time for questions. So if I talk too fast, you can holler. But let me start by sharing my screen. Let's see. All right. So I assume you can see that okay? Great. All right. So Revive and Restore is a California-based nonprofit, but we really work all over the world. And I started this organization now almost 12 years ago with my husband, Stuart Brand. And the whole concept was to actually draw upon the power of biotech, much like you're doing at GMGI right now, but to bring it and make it really actionable in conservation, and clearly some marine, but a lot of it terrestrial. I love this painting because, one, it's by one of my best friends, Isabella Kirkland, but secondly, it really focuses on 60-some species that were all brought back from the brink of extinction. And her website for Isabella Kirkland actually goes into great detail about each and every one of these species. We want to look at how we can apply the tools of biotechnology from sequencing and bioinformatics, advanced reproductive technologies, all for the purpose of ecological restoration. And we're doing that for both endangered species, but also thinking about how to impact extinct species that may still have a place in this world today. Well, how we work is really in kind of 3 unique ways for a nonprofit. We convene workshops all over the world with experts on various different topics. And through those workshops, we start to come up with ways that we could actually really innovate in the field. We advance and launch R&D efforts in labs, again, all over. And we're funding many proof of concept projects, both in the lab and in the field and conservation. Today in the 21st century, we face conservation challenges like none before, from wildlife diseases, invasive species, the loss of genetic diversity, the coral bleaching and other effects of climate change, poaching and wildlife trade. All of these things are completely exacerbated with climate change. And so we think at our organization a lot about how we can facilitate adaptation to many of these threats. We have projects now, some 71 all over the world, focused on 87 different wild species, and we're partnering with 60 institutions to do so, including GMGI, I'm very proud to say. So I'm going to start with something I think close to home for many of you there on the East Coast, and I'm guessing a number of you from the Massachusetts area, and that's the horseshoe crab. Because foundational to a lot of this is going to be genomic sequencing. Starting with the horseshoe crab, though, right there in your backyard, many of you have seen it, and I bet many of you have seen the populations plummet over your lifetime. These horseshoe crabs provide sustenance not only for wildlife, but for all kinds of ecosystem restoration, especially this red-knot bird that migrates as one of the longest migratory birds. This incredible species has been on this planet for 445 million years, and it has survived, as they say, since it is a living fossil, from climate change to asteroids to volcanic eruptions for centuries until humans. And why is it threatened by us so much? Well, number one, it's because of their blue blood, which was identified there in Woods Hole back in the 1970s. And that blue blood, I assume a lot of you know, so I'm going to talk fast through this, has a very unique protein. And that protein, that blood is extracted over 175,000 horseshoe crabs a year from the state of Massachusetts waters are impaled and bled for their unique blue blood. That property allows scientists to use it to detect endotoxins. And those endotoxins have, that type of testing has proved invaluable to the safety of injectable drugs, vaccines, implanted devices for both humans and domestic animals alike. Every time I read a newspaper article on the horseshoe crab, it says they save lives. And that's true, but it doesn't have to be by impaling them and taking their blue blood away. Scientists back in 1995, an incredible husband and wife team there from Singapore, identified that unique protein in their blue blood that allows them to shut down their system when it comes in contact with the toxin. And they were able to actually synthesize that using then state-of-the-art technology of synthetic biology to produce something called recombinant factor C. Now, ever since 1942, we've understood that the safety of medications, injectable medications, would rely on endotoxin testing. And they used to use hundreds of thousands of rabbits for vaccine testing. And until 1995, that blue blood was identified, and the assay is called LAL. And it has been sold by 3 primary companies that mass produce this product for the market for pharmaceutical companies since 1965. It is highly profitable. They pay basically nothing to take this public resource out of the ocean and produce that blue blood and sell it for in very large sums of money. Since 1995, though, scientists have known about this unique property and how it can be synthesized, but it has taken almost 30 years for the US Pharmacopoeia to acknowledge that it is equal to and as effective as the LAL blue blood. Now, at Revive and Restore, this is one example of kind of the advocacy work that we've been doing, is just trying to bring to the public's attention the fact that we don't have to rely on the horseshoe crab to do this. This is the role of the 21st century medication and pharmaceuticals. And what I'm happy to have Jay Bolden from Eli Lilly, who has been an incredible advocate of this. Their company has done more to get the synthetic alternative on the market than anything else. So I'm going to run a quick video by Jay.
I'm a birder, so I have a unique perspective in that I can see the impact of the horseshoe crab not only because of the horseshoe crab itself, but the species that rely on it, including birds and turtles and sport fish and all, anything that's in the ecosystem that's impacted. And so we were aware at Lilly that there was an alternative to the LAL test reagent. We generated the data, but it took a lot of alignment internally, and then externally, we had to convince the FDA and other health authorities around the world. But we did that with our data, and we convinced them. We drew a line in the sand in 2016, and we said that all new drugs that are manufactured at Lilly, we would apply recombinant factor C to them. And so our first product was approved in 2018, and that was a milestone because that was a first in industry approval of any medicine using RFC for release. And then we've subsequently had 6 additional product approvals since then, including our 3 COVID-19 antibodies. Our goal when we set out was to convert all 8 of our manufacturing quality control labs at the time to RFC, and we've done that. And it's been industry is following. We have our peers, Sanofi and Pfizer and Roche Genentech have all come out and made statements similar to what we've done, which is that we'll convert our water testing and all of our new product testing. So there's definitely a movement towards more sustainable endotoxin testing. We look forward to progress everywhere that will make it easier for all companies who have to test for endotoxin to be able to do so using a sustainable alternative to LAL.
So I'd like to just encourage anyone who's in the audience today who would like to support the effort that is right now being reviewed by the U.S. Pharmacopoeia, that would be the only agency that could make the synthetic drug, the synthetic product, I should say, equal to the blood of the horseshoe crab. And it's right now going before the U.S. Pharmacopoeia. November 30th is the last day to send any kind of endorsement or letter of support for it. On our homepage on reviveandrestore.org, you'll be able to see how, if you want to join our petition, we're accompanied by literally hundreds of organizations, Mass Audubon, and a number of organizations right there in your backyard who are really trying to lobby to get this approved. It will be a game changer for conservation once it happens. So I'm going to switch now from my advocacy role to really telling you more that we're doing in terms of funding different projects. And as I mentioned, when we think about this genetic rescue toolkit, we move from sequencing and bioinformatics to cell culturing and advanced reproductive technologies. All of that has to be in place before a lot of genetic engineering can even begin to be pursued. I want to start with the black-footed ferret, which many of you may have heard as part of our story. The black-footed ferret used to live all across the American prairie, from Mexico to the borders of Canada. And basically, it was wiped out early this century by habitat loss and by being replaced by farm and ranch land. But in 1981, once they were thought to have been extinct, but in 1981, one small family of literally just 21 kits were found. And the U.S. Fish and Wildlife and State of Wyoming Fisheries Department brought them in to captivity. And since then, Fish and Wildlife has been reintroducing them quite successfully. They're now on some 30 different reintroduction sites. And it's been a great success story at many levels. However, because only 7 of those, that group of 21, only 7 went on to breed, they are basically a very closed genetic pool, and they are starting to show signs of that inbreeding. So I don't know how many of you have heard of the extinction vortex before, but it's basically a phenomena that happens when their small populations start to lose genetic diversity. They get a lower fertility count, they have a higher mortality, they have an increased risk and susceptibility to disease. And so we were asked by Fish and Wildlife, is there anything that biotech can do to help with the challenges that we're facing with this species? Genetic rescue can, in theory, by bringing in new individuals, really turn the vortex around and increase diversity, increase population stability, and potentially greater resilience to many of the challenges like wildlife disease. Well, we sort of said, well, where do we begin on how are we going to help fish and wildlife with this problem when we heard there were 2 cell lines that had been frozen back in 1981 by the frozen zoo. I'm not sure I'm doing my math right, probably 1985. They had been frozen for 30 some years. Revive and Restore paid for the genomic sequencing on this, and we found 2 cell lines there that had unique genetic diversity. And we thought if we could clone using these cell lines, we could potentially start to reintroduce the equivalent of 2 new founders. We're still in the process of doing that with 1 cell line, but 1 cell line of a female. We worked with Biogen as a cloning company and San Diego Zoo Wildlife Alliance to actually bring this to fruition. And that is Elizabeth Ann. She's the first cloned black-footed ferret. She's the first cloned U.S. endangered species. We now are in the process of cloning a male, and with the goal that once these ferrets start to breed themselves, their offspring will be bringing in new genetic diversity. Now, the one thing that Fish and Wildlife saw as soon as we were underway with this project, and they saw the success of Elizabeth Ann and this beautiful, healthy young ferret, everyone looked around at each other and said, my God, what should we be biobanking and cryopreserving today? I mean, these people were prescient enough to bank that tissue when cloning didn't even exist, right? So what technology is going to be out there in 34 years that we should be doing given the rate of species loss? In the US alone, there are some 1,500 US endangered plants and animals right now, and less than 14% have any level of cryopreservation. And I showed this to the team of we're working with at U.S. Fish and Wildlife, and they could see how dramatic this would be if we could actually start to cryopreserve sooner than later, both to help with management decisions using their genomics and understanding potential inbreeding issues, identifying better management strategies for that, but also for the future. And with that, we've launched quite a significant program. Seth Willey has been the Fish and Wildlife Service Director that we've worked with most closely. Our pilot right now is underway.
We're responsible.
And I'm going to skip that for real quickly because I don't think we have enough time, his video. But what he's going to tell you is that the first one that we were able to sequence and cryopreserve was the Mexican wolf. And what we've been doing is setting up protocols. And this is a collaborative effort. We work with scientists all over that have been from the Smithsonian and other areas who have been doing cryopreservation to come up with protocols that are in a publicly accessible area for conservationists, where they can take just a small specimen, a tissue specimen, so that it can then be cell cultured down. And we then create the roadmap so that it can be sent over to USDA at Fort Collins for long-term storage under all of the supervision of our US government, since they are US endangered species. So we're on a path to do more of that cryopreservation with US Fish and Wildlife. And but I also wanted to show you that in parallel with that, we got quite focused on the coral situation and realized if we could help identify new methodology to cryopreserve coral, it could be a game changer. As we know, the rate of loss on these coral reefs right now is, in some cases, up to 50%. And statistics are telling us trends by 2050, we could lose 90% of our coral reefs. So the pressure is on to do as much as we can to bring them into captivity, both for propagating in living nurseries right now for coral, but also ideally to culture them and to create stem cells and cryopreserved areas. We're working with a scientist at the Smithsonian Institute named Mary Hagedorn, and I'd like to share with you some of her words here.
I've been working on the cryopreservation of marine species since 1993, and I realized it was going to be important because you could tell the oceans were going to be in trouble from climate change. Initially, when I started, I was really just trying to work out the science of how we do this, but then it became very clear that things were changing very rapidly, and climate change and bleaching events were happening. And, I became very worried that we needed to get this science done quickly. I work on the conservation of coral, and I use human fertility techniques to do this. Basically, we are cryopreserving or freezing, however you want to think of it, gametes, the sperm and the larvae of coral, as well as small fragments of coral. And the reason we are interested in using cryopreservation is that it allows us to stop time, really. We can freeze coral and keep them for a day in our bank, or 10 years, or 100 years, or 1,000 years. So it gives us the ability to use this material today or save it for many, many years in the future to potentially reseed the ocean. One of the major outcomes of our process is scalability. We want to be able to cryopreserve coral easily and cheaply so that every professional that wants to around the world can form their own cryopreserved banks of coral and thus speed up the process of securing the biodiversity and genetic diversity of coral. My hope for what we're doing is that these tools, these frozen assets that we're developing with cryopreservation can be used to help for restoration today, but also can be used to help reseed the oceans in the future.
So our effort in coral is quite significant. At Revive and Restore, we have a portfolio of, I think, something like 12 different coral-related projects that we're working with PIs around the world. We're looking at group of the scientists were the first to develop coral stem cells, and we'll be publishing on that later this year. We have scientists working on proteomics, working on some of the major coral diseases, stony coral loss disease, developing assays that will look for stress detectors early on before coral even shows signs of bleaching. So it's an incredible portfolio, and I'm happy to say one of the unique things about the way that we work as a funder is we try to create real collaboration between these different teams in a truly non-competitive way, so that they can almost have sort of sister projects in many ways. And we're really looking at the whole portfolio, not just the success of any one R&D effort. I'm going to stick with the ocean for a while because I know it's relevant to many of you. One of the projects that we've been funding has to do with our kelp forests here on our west coast of the US. I'm based in California, if you didn't get that already. And kelp obviously provides an incredible ecosystem from sea otters all the way down. And fundamental to that those kelp forests is the sea star. And the sea star got hit by an unknown pathogen that is referred to as sea star wasting disease, because that is the horrible effect of this organism. And it has completely depleted kelp forests all up and down the California, Oregon, and Washington border. And we were approached by a group of scientists who said, we have reason to believe that, actually, let me go back on that. We have reason to believe that there are some sea stars that are showing resilience to the pathogen, or some that are rebounding. And entities like the Nature Conservancy have wanted to create nurseries to try to, in captive breeding, reintroduce sea stars. And the scientists said, if we could come up with the genomic knowledge around that, of which are resilient, we could try to, through selective breeding, not genetic engineering in this case, but encourage the release of those and the breeding of those sea stars that are resilient. That work is really important because what is replacing those kelp forests is this purple sea urchin. And there are sea urchin beds that go on for hundreds of miles along our coast. They're absolutely prolific eaters and breeders, and they leave no kelp behind. So this work is really pioneering. It's showing early promise, and it's being headed by Michael Dawson at the University of California at Merced. Over on your coast, we were approached by Andrea Bodner about trying to help with a problem and her team with the Jonah crab. And I'm really proud to say they're closing in on some of the early research that we funded in genomics to understand more about this crab, who used to be just considered an industry bycatch, but is now becoming a very valuable potential resource for the fisheries. And there's the huge potential for overexploitation. So if their chromosome level reference genome is awesome, as I suspect it will be, Andrea, you're going to be able to help inform better management and fishery use there, and to maybe even set up stock quotas and things like that for capture. So we're really excited and confident to see where that is going to go to help with conservation. I'm going to kind of end on one very sort of different approach to all of this, which is the fact that this is Micronesia, and we were approached by a group from the Ulithi Atoll with the challenge of how to integrate biotechnology in many of these cultures that have been managing and conserving their reefs and their ecosystem using indigenous knowledge. And how we intervene in nature with biotech is going to have to become one of the most sensitive and new terrains possible. I mean, we really have to figure out how to do this in a respectful and integrative way. And this group, when they approach us, I felt it really had the beginning of kind of an approach to it that made a lot of sense. So I'm going to let Magul Romal, who's a community leader out there, explain to you some of the challenges.
We're from the atoll of Ulithi. It's part of the Yap state in the chain of what we call the outer islands. And it's basically about a stretch of 99 islands in Micronesia. We're very subsistence community. We depend so much on maybe 90% on our resources, our local resources. So we still grow our own food with very little import. I think like many people, many coastline communities, we are facing climate change. And that's no surprise to everybody, but definitely not for us. We used to be able to predict certain birds and certain fish migrate associated with these weather patterns and those associated stars that we call the traditional markers. Now, these patterns have changed quite a bit, and we can't depend so much on traditional knowledge because it's changed. And so I think that acceptance of that and that willingness to seek further or new information kind of started earlier on. We need to seek more information because it's moving much faster than we ever know. So it takes science, it takes genomics and DNA that is now collected to the way that more accurate count of assessing the reef health and the fish count. It takes that kind of science and knowledge to be able to help us make a decision on management. We've seen positive changes in combining these wisdoms of the West and traditional knowledge and coming up with creative ways to adapt to the fast decline in some areas. I think that the responsibility now in my mind is to spread this to the region, low-lying communities like us, to join the efforts and see from the 10 years of data of how we can and we will be able to fight some of these fast-changing decline in our reefs and resources.
When I think about the changes that have happened over time and the 10,000 years that humans have lived on this planet, it's so obvious, the loss of biodiversity, the loss of bioabundance, and how so often those were the unintended consequences. We didn't really know what we were doing when we were, completely overhunting or exploiting many of these resources until it was too late. But I think a lot about where we're at right now and what we can do for the next 10,000 years. And I believe that by thinking about the intended consequences of what humans can bring about, we can affect change and create a more biodiverse and more bioabundant planet. And that would be turning the tide on species loss. Thank you, Andrea. I think I'm going to let you take it from here.
It's great. Thank you so much, Ryan. It's so interesting. So many different projects that you have going on. So questions are just starting to come in. So let me just pull that up. The first question is, what is one of the most successful reemergence of species achieved through biotechnology?
Well, I do believe that is the black-footed ferret right now. The black-footed ferret, although it's not extinct, it's not bringing it back from extinction, it was as close as you can get. It was thought not once, but twice to have completely disappeared all across the American prairie. And I think that without cloning, there would be no hope that we could turn back that extinction vortex. We had to create, and still have to create, more genetic diversity. Now, the other thing that I didn't mention in the slide presentation, but the black-footed ferret also suffers from something called sylvatic plague, or bubonic plague as it's called in humans. It's very much there across the prairie. It affects all kinds of wildlife. Black-footed ferrets are 100% susceptible to it. And one of the other areas that we're working with in biotech is to identify opportunities to make the ferret more resilient to disease, but also to potentially create the effect of a genetic vaccine, a heritable vaccine against plague. So it's not successful yet. We got a long, long way to go on that one. But I think on the cloning, it's a major win for conservation.
That's great. Maybe following on from that, the question is, we're learning that our microbiomes have tremendous impact on our health and fitness. Instead of altering genes of the host, is it possible to alter the microbiome to make a wild animal more disease resistant?
I'm glad to get the microbiome question, because one of the projects that we're funding with coral, it will be and is looking at the microbiome of the coral and the symbiont. We probably have not paid as much attention to the microbiome as we can, because at the genetic level, in many of these situations, we're dealing with a surrogate species. In the case of the black-footed ferret, we were dealing with a domestic ferret, which has its own microbiome as the surrogate mother. The other clone that we've done is with the wild horse, the only true wild horse in the world. We cloned the Przewalski's horse, and we used a domestic quarter horse mare. Her microbiome is the one that would be gestating the Przewalski's horse, and we've seen, we've seen no negative impact from that combination. And but this is a really good research area and one that we are pursuing.
Great. Here's a question from Mary Moore, who is asking, how time and resource intensive is cryo work, or the cryo process? For corals, for example, is your approach geared more towards preserving as many genomes as possible, or focusing on more favorable genomes?
Well, we don't know which are the favorable genomes. That's the toughest question. There are very few corals that have full genomes sequenced. So I think people like Mary Hagedorn, the Smithsonian scientist that was featured here, her interest is in ensuring that there's samples of everything possible while they're still alive, because many of these reefs are being wiped out. So the cryopreservation, to develop a cell culture can be timely. For mammals, the wolf and things like that, those were done, I believe, in 2 to 4 weeks. They take that living tissue and they create a cell culture medium. They verify that those cells are dividing and expanding appropriately, and then they cryopreserve. That's pretty fast-tracked. Mammals are relatively easy. Other species, amphibians, it has taken almost a year for some of the researchers to cryopreserve amphibian tissues because of all the contamination of skin surfaces. So it really depends on the taxa, and unfortunately, it's truly complicated and needs a lot of R&D effort. And we're trying to raise funding for cutting edge research on cryopreservation of these hard to cryopreserve species.
In addition to corals, do you know of any efforts, or are you supporting efforts to biobank other endangered or threatened marine organisms?
Yeah, I think that fish in particular have had some of the greatest progress in cryopreserving sperm. That's relatively easy, and I do believe that the fisheries have a lot of cryopreserved material from, salmon, trout, etc. Where it gets harder are on all these less known fish that do not have fan clubs like salmon. And so I believe the current statistic is that less than 11% of fishes in US waters have ever been cryopreserved. So it's a fraction. And that is an area we want to move into too, funding dependent.
Actually, there's a following question from Ian Gardner to ask if you have been in contact with Ocean Genome Legacy, which is a biobank at Northeastern University in Nahant, where they have cryopreserved DNA and tissue samples from a lot of marine species.
Yeah, I have. I was lucky enough to tour that facility a few years ago. And I think one of the difference, if I recall this correctly, and so don't get me wrong, is that we're trying to create cell cultures as well. So living cells, not just cryopreserved archival material. And the only reason we could do what we did with the black-footed ferret is because there were living tissues. So, but we need more entities like Ocean Genomics Legacy and others doing this kind of cryopreservation work at every level.
Thank you. Beth Gettle asks a question: would biotech be able to help the effort working against beech leaf disease? Here in Pennsylvania, we're losing beech trees to a nematode that is causing disease.
Oh boy. Well, the big advance in resistance if we go back to trees, the biggest and best example of engineering with biotech for plant health is with the American chestnut tree. And the whole concept there, which many of you may know about, has taken some 30 plus years of selective breeding and backbreeding and genetic engineering to create a blight-resistant American chestnut tree. Unfortunately, just in the last 6 months, they've acknowledged that they've had some setbacks in the success of that effort. And depending who you talk with, they point to different errors that may have occurred or different challenges. I was so hoping that tree, and I still hope that tree will open a door to other trees that are suffering from pathogens, because there are so many out there. And we don't know of any other approach right now on defeating those kinds of wildlife diseases for plants that are a better success story. So I'm still, I'm still rooting for the American chestnut tree success.
An anonymous question: do you get any political backlash about the cloning programs? And if so, how do you get past that?
I think I wouldn't call it backlash. I would say that people have questioned whether or not any of this technology should be used to intervene in nature versus letting nature take its course. And that one, I feel very comfortable answering, which is that I don't think it should be a zero-sum game. I believe that we have to do everything we can to protect the species that are there, to help them prevail. And I don't think it has to be either or. So, I think that the way that science has advanced in human health, incrementally for many of us, we still lose family and friends from all kinds of horrible cancers and heart diseases and everything else. But it's a lot different than it was a century ago. And it's only been through this incremental progress that we've had in science that we've been able to protect the lives that we have. I think we have to do the same thing for wildlife. So that's how I answer it.
That's a very good answer. It seems like each organism may need a different approach and may have different levels of risks and different unintended consequences. Is it possible to use advances in computational modeling and AI to evaluate risk, predict outcomes, and to increase the throughput of this approach?
Yeah, I'm excited about AI for exactly this reason. So I think there's a lot of ways that we can do better modeling. I'm if there's anyone on this call or scientists that are in your field that are working in this area, I would welcome it. We've had a number of discussions in some of our workshops about the use of AI productively to advance this area to lend more insight into outcomes. But we don't have yet a funded project in that area, and I'd love to.
All right, a question from Ella Show, who says, I'm a student at the Gloucester Biotechnology Academy, and I'm wondering what accomplishment of your company you are most proud of.
Well, first of all, I got to tell you, it's not a company. This is a not-for-profit organization. And I think I wouldn't point to one project. I think what I would point to is the fact that entities like U.S. Fish and Wildlife are actually partnering with Revive and Restore. That they're actually saying, help us think through how can we innovate in this field. It takes a lot in the traditional field of conservation to think outside the box. Traditional conservation has focused on preserving habitat and removing some of the invasive species or whatever it is, but they haven't really thought about using biotech. And so the fact that Revive and Restore has influenced, in just a decade of time, has influenced conservation leaders from both marine and terrestrial conservation is, I think, the greatest inroad I could have ever hoped for. Because at the end of the day, we're not trying to create a zoo curiosity. This is not about, the one cool species. It's really about integrating these technologies as a complement to the existing conservation toolkit.
So maybe a follow-on question to that is how does Revive and Restore prioritize projects?
In a couple of ways. It was fun. I was just working with our program managers on criteria. Which projects do we just select and why? And sometimes we'll do a very specific call for proposals. We did that on what we called wild genomes, where we said, tell us about the conservation problem you face and how having just a genome, having a reference genome would actually impact conservation. Andrea gave us a great proposal that said it's going to, technically impact management decisions. The sea star wasting disease guy said, look, we're going to be able to selectively breed better, more resilient sea stars. We look at how compelling is the proposal. We look at how compelling is the team to pull it off. We look at geographic distribution, West Coast, East Coast. We look at a lot of different factors. We just funded a project in Kenya that is setting up the first genome lab for helping to identify forest elephants that, as they say, are invisible elephants. It's so forested, they don't know even how many there are. But guess what? Every elephant at a certain time of year goes to the same watering hole. And what do? They poop. And that fecal matter can be, with eDNA, an insight without a camera, without a lens, of identifying how many unique elephants are in that forest. And that lab is being run by local Kenyan, very early on scientists. And it's so exciting. So, we go from the forest to the sea, and different countries trying to increase diversity, and all of those things that we can do with, budgets that are always dependent on the funders and any constraints we have to agree to, around the funds we collect.
That's great. There's a question here from Allie Cook, and Allie is writing in from a watch party at Gordon College. I guess the students all get together and watch the Science Hour together. And Allie's question is, do you have any efforts currently going into other severely bottlenecked species, such as the leopard seal population?
No, somebody should send us a proposal. But yeah, we look at species that have limited populations all the time. But that's not one that's on my radar screen. And again, compelling proposal. What difference will it make? We always ask the question, what will this information do? What approach are they taking as conservationists that will be a game changer for the species? Species like the porpoise. Sorry, all of a sudden I'm blanking on the name. The tiniest of all the porpoises.
The vaquita.
The vaquita, thank you. The vaquita has been very hard to cryopreserve. I mean, they finally got samples. Just handling those porpoises causes, a heart attack. So how can you deal with some of these small, vulnerable populations without being invasive? So, some species are going to be easier for us to have an impact on, and we welcome those opportunities from everyone.
Great, thank you. Here's a question from Emma, who also appears to be part of the Gordon College group. If the ferret population declining due to lack of genetic diversity could be theoretically combated by introducing cloned ferrets to the population, how many cloned ferrets would you have to introduce in order to reverse the vortex you mentioned?
Really good question. And if we had that AI modeling, it would probably be better for me to answer that, but we don't. But the U.S. Fish and Wildlife Service has been breeding animals now for 30 years. And the good news is they're rapid. They're very prolific breeders. The gestation time is literally months, and their average lifespan is 3.5 years. So we wouldn't be necessarily the clones that we're producing, it'll be their offspring, and they will only be introduced after those offspring have proven to be fecund and, carrying no deleterious mutations. So we've got a ways to go, but the modeling would be done based on those offspring. And I believe over time that we could get up the numbers enough to actually start to impact diversity.
All right, just maybe a couple more before we run out of time this evening. How are CRISPR technologies that have the ability to make very precise changes to DNA having an impact on biotechnology for conservation?
Not nearly enough yet. Even the American chestnut tree, which has been a pioneer in engineering, did not use CRISPR technology. So I welcome the advent of it. Some of the projects that we're working with invasive species, with mosquitoes and things like that, are starting to use CRISPR technology. I think that'll be probably one of the first uses. The sylvatic plague that I talked about with the black-footed ferret, we're talking with scientists that would be using a CRISPR-based technology to do multi-gene edits. But right now, we don't have anything underway advancing that technology, but it's going to be in the works.
All right, a question from Bonnie Fenrock. Are there any aspects of your work that can slow the progress of climate change, or are you mostly mitigating the results of climate change?
Right now, all these projects are around mitigation in that sense. There are efforts that we've looked at that have nothing to do right now with Revive and Restore, but are ways of sequestering more carbon in plants, rapid growth areas. There's a number of different ways that conservation could be applied to this, but most of those are more commercial operations that are trying using both kelp and other marine organisms for sequestering carbon. They're not nonprofits, and they're able to kind of draw upon those investment funds that are being set up for climate offsets.
All right, and just one last question before we wrap up the evening, and it comes again from Mary Moore. Where do you hope to see Revive and Restore in the next 5 years?
Yeah, so I love that question because I'm working on my 5-year plan with the team, and we basically have all these different channels. So as I showed you that genetic rescue toolkit, we want to be able to have R&D efforts in each of those areas, achieving some impact that I can be showing you of what is the most, what project are we most proud of this year. We'll have more and more examples of in the field application of these technologies. But we look at it in portfolios. So there'll be advances in reproductive technology for birds. You can't clone a bird like you can clone a mammal. And we have a significant effort underway led by Ben Novak. We've got 8 different teams around the world working on primordial germ cells for birds. That'll be a game changer for endangered birds when the time comes. So I hope that we have success all along the way, including in gene editing. And stay tuned.
Wonderful. It's a great way to wrap up the evening. So thank you so much, Ryan, for generously sharing your time with us, your insights, and your really amazing stories. And thanks to everyone in the audience for joining us this evening. Apologies that we didn't get to everybody's questions. There were a lot of questions that came through this evening, but we'll hope that everybody will join us again for the next Science Hour, which is Thursday, February 29th, where we will host Jeff Kneebone, a senior scientist in Fisheries and Emerging Technologies program at the New England Aquarium. And he will discuss his research using electronic tagging and eDNA to better understand the life of marine fishes. So please stay tuned, stay in touch. We love hearing from you, and we look forward to seeing you again, either virtually or in person at our beautiful research facility on Gloucester Harbor. So thanks again, Ryan, and good night, everybody.
Thank you.
The Gloucester Marine